polarized. They come over to the side that's exposed to the T cell. And the T cell has lipid rafts with the TCR and the CD4, let's say, and the CD3. Those lipid rafts have a tendency to come over onto the area that's exposed to the APC, so that those two cells have a large number of adhesion molecules that bind at that synapse, that immunosynapse. There's polarization. And your book has some interesting photographs of that. Things actually will migrate to that interface so that those two cells are firmly docked together. So you need to know what an immunological synapse is. That's the docking area between two immunocompetent cells, APC and T cell. And then I want you to know what polarization is. That's where those lipid rafts, those adhesion molecules, migrate to that synapse. Okay, a little bit more detail on the surface antibody. Here we have surface antibody. It can be IgM or IgD. Your book likes to call it B cell receptor, but I like surface antibody. The surface antibody, that membrane gene segment, just codes for a little hydrophobic region here that allows the peptides, the heavy chains, to be anchored. There's not a big extension into the cytoplasm. But there's co-receptors that bind to that heavy chain. This is the Ig alpha, and this is the Ig beta. And if you notice, they are linked to each other with a disulfide bond. This is an immunoglobulin-like domain. It has those beta strands that we've talked about. I just gave you part of an answer. Anyway, here's this cytoplasmic extension. These are called ITAMs, and your book likes to have them noted as being yellow. ITAMs are extensions into the cytoplasm that's present on these receptors or co-receptors. And ITAMs are immunoreceptor tyrosine-based activation motifs. Aren't you glad we're going to use the acronym? ITAM. These tyrosine residues are phosphorylated by kinases when the receptor is bound. Okay, so that can help, that facilitates, accommodates transduction. So these ITAMs are important, and we're going to go through some examples. But the important thing here is to note that the heavy chain doesn't have an ITAM. It's on the co-receptor, on the Ig alpha and Ig beta. And those must be close to that surface antibody. Okay, here's the T cell receptor. Again, here's the peptides, alpha and beta. Let's not worry about gamma and delta. Are any ITAMs on alpha and beta? No. But here's a whole cluster of peptides that are usually associated with the T cell receptor. I always think, surface antibody and the T cell receptors just get lonely. They need to have a cluster of other peptides around them. If you notice, this whole structure is called the CD3. CD3. And it contains gamma, delta, 2 epsilon peptides, and in this example has 2 zetas. Okay? There's also an eta that can be present instead of the zetas. So I know this is pushing our Greek alphabet terminology here, but that's all it's, that's in sequence. Alpha, beta, gamma, delta, epsilon, eta, zeta is how it goes, okay? These, you always have these paired together and those paired together and then 2 paired like this. Notice all of these in the CD3 have ITAMs, right? These are phosphorylated when the TCR binds the MHC peptide structure. And you can see this is a nice layout of how they're configuring. Don't worry about that. The ZAP70 is a signaling protein. It can bind the phosphorylated ITAM. So here's an ITAM that is phosphorylated, and then this ZAP70 can bind it. This ZAP70 has a kinase domain. So can you see where we're going from here? The ITAM is phosphorylated, and then this ZAP70 or something like it can bind it, and then you have a kinase present right there. Okay, you don't have this slide, I suspect. Do you not have this one? Okay, so this shows an immunosynapse. I tried to explain this earlier. This is the interface between the APC and the T cell. That's the synapse. And this is a fluorescently labeled photomicrograph where they have localized different adhesion molecules. And you can see that the ones that they've labeled are in that synapse. They've all polarized to that interface between the two cells. So polarization is pretty well understood. That happens. And that way you have a lot of adhesion molecules linking those two cells together to accommodate the docking. Okay. For activation, you need about 50 TCRs bound to MHC antigens in order to activate that T cell. For a B cell, you need over 20 B cell receptor surface antibodies bound to antigen. And typically the antigen is best if it's multivalent so that you have cross-linkage of those surface antibodies, where one antigen is bound by 2 or more surface antibody molecules. I have a picture of that coming up. But what happens during this activation process, okay, here we have TCR, right? The MHC, class 2 peptide, the TCR. And CD3 right here, oh, it's bound, so all of these are phosphorylated. We have the CD4 bound here too, and it has this lick R, this lick protein, LCK. This is a receptor-associated kinase. It's on the cytoplasmic face of the CD4 and the CD8. It's activated when that co-receptor, when the CD4 binds the MHC, and that LCK is what adds the phosphates to the ITAMs. So this is all of these, the TCR, the CD3, and the CD4 in this case, are all on the same lipid raft. When this is bound, when the TCR binds, and the CD4 also binds, that activates this lick, and it's going to LCK, I should say, this LCK, and it phosphorylates these ITAMs. Isn't that neat? And then the ZAP70 can bind those. That's another kinase. And that other kind, this goes into the mechanism. I don't care that you know about that. Don't worry about this. This ZAP70 then adds phosphates to scaffolding proteins, LAT and F. Don't worry about this. But in essence, what happens after this ZAP70 adds phosphates to scaffold proteins, you have the activation of the phospholipase C, the PLC. That's an enzyme that forms two important cytoplasmic signals, inositol 3-phosphate, or IP3, and diacylglycerol, which is DAG. Now, I hope you've talked about this in cell biology. I know I talked about it in biochem. These are two important signals. The phospholipase C makes IP3 and DAG. Okay? I think the next slide goes into IP3 opens calcium ion channels in the ER. Calcium ions can also come in through the plasma membrane, but it opens up. The endoplasmic reticulum stores calcium ions, and IP3 will open up those ion channels. So the calcium ions will flow from the endoplasmic reticulum into the cytoplasm. Once they are there, they can activate phosphatase and calcineurin, but I'm not very familiar with this. Calmodulin is a regulatory protein that's activated when it's bound by calcium ions, and it in turn activates other things. So what's happening is that we have the TCR binding the MHC peptide. We have the CD4 binding the MHC, and that LCK is activated. It phosphorylates the ITAMs. The ITAMs are then bound by the ZAP70, and it in turn activates other things that in turn activate this phospholipase C, which makes IP3 and DAG, and now we have calcium ions coming in. And the calcium ions activate other things, and the calmodulin specifically can activate a transcription factor called NFAT. NFAT, I like that. I identify with that. NFAT. So transcription factors activated. Okay, DAG. DAG, don't worry about this recruits, these RAS proteins. These are G proteins. DAG activates a MAP kinase cascade, a whole cascade of things that eventually activates protein kinase C, which adds phosphate to other proteins, and this results in a different transcription factor being activated, NF kappa delta. So what happens is that you have cascades being activated when these receptors are bound. And you have a number of different signals that are made along the way. The main end result is transcription factors are activated. Now, the ones that I really want you to know, I've underlined. You know, the ZAP70, the LCK, ZAP70, phospholipase C, because it makes both IP3 and DAG, which I think you should know. IP3 allows calcium ions to come in. You have to know the whole cascade. No, not everything. We've skipped over some. But the transcription factor. DAG activates protein kinase C. That's the important one. And you have a transcription factor. Okay. Now, we did skip over some things. The new research is going into the details on how those things are activated, how they bind to each other and so forth. Why? Why is it important to know that? To know how you could manipulate that if you needed to. How could you mess up this activation process if you needed to? How could you activate it if you needed to? How could you deactivate it if you needed to? This shows the whole process for the TCR in a nutshell. Don't memorize this. Please don't memorize this. But it shows one whole, a flow sheet, a one-figure flow sheet. And you can see this is all, I didn't go in the fin and all of those, but this is in a lipid raft. And it just, here it goes through different ways that you can have transcription factors activated. Okay? You won't be quizzed on, there won't be a lot of points on, this will not be the essay question on the next exam. You will not be quizzed. And I think I even asked this on the quiz, on the take-home quiz, or something like that, yeah. I just gave you another answer. B cell activation, how's it different? Well, it's not, the receptor's different. You have surface antibody, okay? Surface antibody. And this shows the cross-linking. This is the most effective type of activation, is when you have a multivalent antigen and more than one surface antibody molecules bound to it. This is called cross-linking the surface antibody. When that happens, then the Ig beta and Ig alpha ITAMs are phosphorylated by these three different associated kinases.